Okay, so welcome everyone to the second day of this virtual conference, the AIRR Community Special Event, Response to COVID-19. So today we will be talking about B-cell receptor repertoire data and how those data may inform our understanding of the pathophysiology of COVID-19 with implications for diagnostics, therapeutics, and/or vaccines. So I'm going to give a brief introduction, and then we're going to have 4 speakers. So we will have first myself talking really at a kind of high level view of antibody responses in SARS-CoV-2 infection. And there we're really going to be looking kind of more globally at the antibody and B-cell response. Then our second speaker, Charlotte Dean from the University of Oxford, will be talking about building databases and tools to aid in measuring vaccine evaluations. And this will really be very exciting for the AIRR community to hear from Dr. Dean about the database development work that she's been doing. And I think it's very much consistent with our principles of sharing data and standardizing data across the world in this effort to fight this global pandemic. Our third speaker, kind of along the same lines in terms of sharing methodology and expertise, is Kenneth Hain. I'm sorry, I'm still saying your name wrong, from Yale University. And he works with Steve Kleinstein on the analysis of B-cell lineages and selection of B-cells in immune responses, and has turned that expertise to a very interesting analysis of COVID-19. And then last but not least, our fourth speaker is Brian Briney from Scripps Research, and he will be talking about isolation of neutralizing antibodies against SARS-CoV-2. And Brian is part of an incredible effort that has generated a massive number of these neutralizing antibodies. And so I think it'll be a very nice way for us to end our session in terms of the speakers. Then we will have a moderated panel discussion where we'll have the 4 speakers provide answers to some pre-prepared questions, but we also want this session to be very interactive. So in terms of it being interactive, we'd like to have participation from the audience, both with respect to asking questions of the speakers, and we've factored in time for that. So each speaker will have 23 minutes for their talk, followed by 7 minutes for questions. Now, the audience members, you can ask questions in one of 2 ways. You can either raise your hand, and then that will give you access to speak, and we'll let you ask a question. Or if you prefer, you can use the Q&A, and then you can type your question. Now, what I would, obviously, if there are a lot of people asking questions, we may not get to all of them, but we will certainly try to get to as many as we can. And if your question wasn't answered right after the speaker's talk, we may use it in the panel discussion later. When you verbally ask a question, please make sure you identify yourself. And if you're asking it of a specific speaker during the panel discussion, please mention the speaker's name as well if you're trying to target the question to a particular speaker. Okay, so with that, I think we're basically ready to go. The other thing I wanted to mention, just in terms of housekeeping, is that there is going to be a 10-minute break. This will be roughly at 12:45 Eastern Standard Time. Now, if the speakers, from the first 3 speakers, including myself, get finished a little bit early, then that break will be a little bit longer. And then we'll start with speaker 4 at the scheduled time that's in the program. Okay, so with that, are there any other comments from the panelists, or Pam, do you have any other further housekeeping that you'd like to mention before we get started?
I would just mention, just a reminder that this is being recorded.
Yes, thank you, Pam, for that. Okay, so JP, if you could give me access to share my screen.